My name is Dan Freeman. I'm the Chief Technology Officer at terraFlow Bio, and today I'm going to be talking about our work to map the long tail of disease immunophenotypes, what 1 million papers teach us about flow. Last year, I presented our platform for deep immunophenotyping. Flow analysis typically consists of somebody sitting in front of a computer and manually annotating cell types of interest, which can be limiting with larger discovery datasets. At terraFlow, we've developed a unique approach that goes into your data, looks at every possible combination of positive and negative markers, and pulls out the key phenotypes that separate your patient groups. In some cases, these may be major cell types you're already gating on, but in other cases, it can pull out these rare subsets that maybe you wouldn't think to look at, but that just emerge from your data as a very strong predictor. Our customers have used this approach to develop novel biomarkers in settings like pancreatic cancer, immunotherapy, Hodgkin's disease that have subsequently validated in downstream testing. Now, there's a lot more I can say about this approach, but today I want to talk about 3 questions that remain after this type of analysis and are a fundamental limitation of any unbiased approach. The first question is, what do these phenotypes mean biologically? Besides being a statistical correlate, is there some rationale that can justify further experimentation? Second, what phenotypes don't appear in my data? This is especially true in settings like drug safety, where you actually want to demonstrate the lack of an immune response, which means knowing a priori what you want to measure. And then the last is that now that I have my phenotype, what do I do next? What's my next experiment? These questions are compounded by 2 trends in the literature. First, the number of papers has been steadily increasing, particularly in domains like immuno-oncology and COVID-19. Last year, there were 150,000 papers that included flow cytometry results, which comes out to about 5 publications per hour. The second is that our panel size has gotten larger. So on the right, I'm plotting the average panel size over time, stratified by decile. So you can see that the smaller panels haven't changed. There's still plenty of people doing 4 and 5 color flow. But it's also become quite mainstream to do 10 to 20, even 30, 40, and 50 color flow cytometry, which means there are thousands of new populations that we're able to consistently measure for the first time. And that brings me to the main finding I wanted to convey, which is the long tail of disease immunophenotypes. So here I'm plotting the distribution of phenotypes by the number of times they appear in the literature. On the top left, we have phenotypes that are almost universally measured in flow cytometry literature. These are your Tregs, your CTLs, your helper T cells. In the middle, we have populations that apply to specific domains of immunology. These are transitional B cells, unconventional monocytes, regulatory CD8 Tregs. And on the right, we have a long tail of thousands and thousands of unnamed phenotypes that represent the biggest opportunity for novel biomarker discovery. I highlight some examples here. There's a subset of Tregs that lack CD25. They accumulate in the joints of arthritis patients and lungs of TB patients, and have been thought to be a strategic reserve that we can tap into using immunotherapy. There's a subset of macrophages in the gut that express CD4 and have been shown to be closely linked with prostate cancer survival. In our own research, we've identified a subset of dysregulated T cells that co-express both GITR and PD-1, and that have since been developed as a target of a bispecific antibody therapy. Now, I'm not saying everything you read in the literature is correct, but if I found a phenotype that perfectly stratified my patients, and there was somebody out there that previously described the exact same population, I would want to know that. And I would want my software to tell me that. And so in the last section, I'm going to show you what terraFlow is doing to bring this dark immunology to light. For the last 6 months, we have been using large language models to systematically map every single phenotype reported in the flow literature. And I'm going to show you exactly how we do that. We start by mining full-text articles from sources like bioRxiv and the PMC Open Access subset. We then feed each passage into a large language model and have it output the minimal string needed to capture that phenotype. We then take each marker in that string and convert it into its UniProt ID. That leaves us with a universal identifier that we can map back to that population no matter how else those cells or those proteins might be described in the literature. We then do the same thing on the disease side using mesh ontologies and capture the coincidence of phenotype and disease in a machine-readable database. As we're doing this, we're also tracking how these seemingly one-off observations recur across diseases and across immune compartments, allowing us to make connections between seemingly disparate domains of immunological research. What this means for you is that when you upload a dataset to terraFlow, we're going to take every phenotype we mine from the literature and recreate it in your own dataset. So at the end of the day, we're able to say, here's what we found in our unbiased analysis. Here's what we expected to find based on the literature. Here's the overlap. Here's where they diverge. And you are empowered to make the final judgment. I close with my vision for flow analysis as a conduit. What I mean is on the left, we have the biological material we collect from patients, which is tied to individuals with various diseases, comorbidities, and identities. And on the right, we have all published human knowledge. And the more events flow through our conduit, the more clearly these paths emerge as clear explanatory variables or as gaps for new biological discovery. The hardware is already built to support this kind of analysis, but the software needs to catch up. And the only way we do that is by incorporating real biological knowledge into the analysis itself. I originally presented this at CYTO 2025. We are actively building this future at terraFlow, and I'm excited to keep sharing this journey with you. Thank you.